So my name is Michael Klutstein, and I am a professor at the Institute of Biomedical Research, Biomedical and Oral Research here at the Hebrew University. And my research basically spans one interesting aspect of biology, and this is how biological systems react to the passage of time. And we do this in mainly two aspects. One is aging, how age affects biological systems, and especially reproduction. That's one of our specialties. The other aspect is related to the current project and is how cells react to a long time without nutrients or with a lot of stress. But we're not talking about long time, 1 day or 1 month even. We're talking about years. How cells react to years without nutrients. What we started to be interested in is we know for many years that bacteria react to long-term starvation with things that they do. For example, they are more resistant to other stresses, or they elevate the mutation rate, the rate at which they accumulate changes to their DNA sequence. But we wanted to know if also more elevated creatures than bacteria, more evolutionary advanced creatures like us, like cells with a nucleus, also react in the same way. So one very simple model of a cell with a nucleus is yeast, that we also meet in our very common day-to-day life, in bread and in wine and in beer. So we took yeast and we starved them for 2 years. And then we looked at what happens to the yeast cells with starvation. And very surprisingly, they have very similar traits to bacteria. They do kind of the same thing. There's small differences, but in the principles are very, very similar. And so we think that any cell that is starved for a very long time will react in the same way. And that opens the door to many exciting thoughts and discoveries. So this all started with a crazy brainstorming that Ronen Hazan and also another member of the team, Itai Gutman, who is a master brewer, had one day in the brewery. And then they started talking to me because they knew I was working on yeast. And then we had kind of a brainstorming session, and we thought it's a good idea to try this thing. And at first we didn't know how to get these archaeological vessels. And then we very naively wrote to archaeologists just a formal letter. And we were very, very happy to see that archaeologists are actually very open to new ideas. And then they just really wanted to collaborate with us. And that's how this whole project started, and the Ancient Beer project was born. And from then, it has been 5 years, and we continue to work together and find very exciting things.
My name is Ronen Hazan, and I'm a professor in the Hebrew University. I'm a microbiologist, and my lab is focusing on treating antibiotic-resistant bacteria. And we have a new method to treat them using phages, which are viruses of bacteria that are very efficient. However, I always dreamed to be an archaeologist. And now I have a very nice opportunity to combine these two passions of mine, to be archaeologist and biologist. So I met a guy who's expert in beer. His name is Itai Gutman, and he started to ask me questions about those phages that we work on, and maybe we can use them to decontaminate beer from pathogens. And slowly we start to talk about many things, and then I found out that actually Itai is preparing ancient beer from wheat, from the uncultured wheat. And then we continue to start talking, and then suddenly an idea that came probably from too much of an alcohol at that evening was that probably those yeasts that were introduced to the vessels when the ancient people made beer, probably those yeasts are still there, because they are invading a species, and there's no reason for them to go away. If they got once in a while nutrients, once in a while water, they are probably there. So this was a very, very far away idea. However, we say, why not to check? why not to try and see if it's feasible. We approached to archaeologists, and they were much enthusiastic than us. We were very skeptic about it, and they supplied us with many vessels. Some vessels are Philistines and Egyptian, some of them 3,000 years ago to 5,000 years ago. We took those vessels, and to our surprise, we succeeded to isolate yeast from them and to make beer from those yeast. Nevertheless, the million-dollar question is, of course, are these yeast authentic? Are these yeast really the descendants of those yeasts that were used by those Philistines to make the beer? So to this end, what we did was to take a lot of controls, a lot of vessels that are unrelated. For example, lamps like this one. They for sure didn't have a beer in them. And we tried to see if we see yeast. And again, very surprising, we did not find any yeast. I will add also that the yeast that we found were related to beer and to wine and to other materials, and they know how to live with wine, or with alcohol, which means that probably once they saw this and they are adapted to that evolutionary. So we get many evidence that those yeast are actually authentic. Since then, we develop a lot with our research, and we are progressing it to many directions, including more of a DNA direction, looking at the DNA of those yeast, looking at the DNA of any microorganisms that is within those vessels. And also we are making those beers that we think are very exciting to drink them, because they most probably have the taste of the ancient beers.
How long can yeast really survive without food? And to ask this question, we took it to the most extreme case where we could think about it, and this is thousands of years without nutrients. And how can we find yeast that was starved for so many years? And to do that, we went to archaeological vessels that from thousands of years ago, when they contained beer thousands of years ago, and then they were starved for many, many years. And together with my dear colleague, Professor Ronen Hazan, we developed this pipeline that is able to revive these organisms, these yeast, these dormant cells that are starved for so much time, and characterize them and revive them and grow them and even remake beer out of these organisms.
So my name is Shunit Glazer, and I'm the lab manager in Professor Ronen Hazan's lab. We receive ancient vessels from archaeologists, and our students take them and essentially imbue them with media that is good for yeast, and that enables the yeast to propagate inside, and we sort of try to enrich for yeast and inhibit bacteria, because we get a lot of soil and we get a lot of microorganisms growing there. And what we do later is try to isolate the yeast. We streak them on plates, and then we view them in the microscope to make sure that we're talking about yeast. And then we try to characterize these yeast, essentially to try and find out what we found. Did we find something that belongs to soil? Did we find something that belongs anywhere else? And the second we get the yeast, what we do is sequence them and try to see if they belong to yeast that can grow with alcohol, or belong to beer or wine or oil, etc., or just environmental yeast. Well, we work with the archaeologists, and they essentially tell us what vessels are predicted to be. And we try to use these vessels and collect the samples from those vessels, and we get a lot of other samples as controls. We also use people who help us with the bioinformatics, and we have a lot of collaborations here. And essentially at the end, what we do is try to make beer, and for this we have beerologists that help us with that.
So I think that one of the reasons to be archaeologists, one of the reasons that we're so curious about the past, is actually that we want to feel what they feel. We want actually to experience the past, to better understand it. And there's no, I think, a better way than to eat or to drink the ancient food to understand that. So this is called experimental archaeology, where we try to reconstruct the past, we try to make food again, we try to simulate the drink. For sure, this is not what was there. It's probably different a little bit. However, we are very close because we are using the same yeast that they used to use. And I will add, these yeast are not ancient. However, they are the descendants of those yeast. So we term this yeast heritage yeast because those yeast they are not the original yeast that lived 5,000 years ago, for sure. They are the descendants of those yeast, and they keep the same phenotype, the same characterization of the ancient yeast, and nowadays we can use them to make a new beer and to see what was the taste of the beer at that time.
So the idea of gas comes from the many years ago in the 70s, very important microbiologists who were working on bacteria and were thinking about this phenomenon in bacteria. And there it has been really extensively studied and shown, and it has a very important role in bacteria because the bacterial culture really never really dies. It never dies completely. It will always be cells remaining that stay alive. But the transition of GASP from the bacteria into eukaryotes, into cells with a nucleus, this is a new idea that we are currently exploring in the lab. So GASP is initials for growth advantage in stationary phase, and that was the scientific name given to the reaction of bacteria for a very long-term starvation. And the main things that happen to bacteria in this very, very long-term starvation is that they accumulate resistance to, very strong resistance to other stresses. So, for example, if you starve yeast for a very long time, they will be more resistant than other bacteria to a shock of heat stress, for example, or to more acid. They will be more resistant and will grow better in these conditions. That's one thing. The other thing is that they elevate the mutation rate in their DNA. The reason for that is that the continued stress enables an advantage to change the genome very frequently. So but they basically stop to repair and damage to their DNA, and then they accumulate changes, mutations and changes to the DNA sequence, and then the variation within the community of the bacteria which are starved grows, and the selection, the evolutionary selection, becomes much, much more efficient and much easier. So also, another point is that basically the GASP, if you think about it really deeply, is very, very common in nature. Actually, it's the most common situation for cells in nature. Most of cells in the natural environment are actually starved and not eating. The way that we feed microorganisms in the laboratory setting is that we really overfeed them. And for example, in seawater, where there is a lot of bacteria, there is very little nutrients for these bacteria. So if you add nutrients to seawater, you see immediately that the microorganisms start to reproduce, and we have much, much more. So the more common situation in nature is actually this situation that we are studying, the GASP and the long-term starvation. The findings have very exciting possibilities, both in terms of microorganism behavior, but also for higher creatures. So let's start with the microorganisms. The fact that we can actually find microorganisms that survive for so many years enables us to perform archaeological research using these microorganisms. And the makeup of ancient vessels or very old pottery that contains bacteria and fungi, and to characterize which fungi and bacteria were there will give us another angle to archaeological studies and will enable us, for example, to identify what was the use of the original use of this vessel. So that's one aspect. But actually the phenomenon is much broader than that. And if we know that cells can survive very, very long periods of time without getting nutrients, then, for example, space travel is possible for these cells. And we would like in the future to develop some time capsule which is able to close these cells for thousands of years and send them somewhere very, very far away. So that's one use. But the most amazing thing, I think, for this research is that we actually found that even cancer cells can survive many months without any nutrition. And the question is, what does it do to the cancer cells? And we have reasons to believe that this very, very long-term starvation that we have for cancer cells actually makes them more aggressive. And that this is something which happens in cancer tumors all the time. So there is a problem. I mean, a lot of approaches to cancer today, for example, deal with nutrition and say that it's better not to eat a lot or to eat much less calories during recovery from cancer. It's not very clear that this is good, because then you are kind of starving the cells and you're creating this starvation situation. So this is all still under investigation. We still don't really understand what's going on, but we know today that the starvation of cancer cells is something that happens in the tumor and has a role.
So I see it as another tool for archaeologists to bring the past to the present. Our aim is actually to help them to understand better the past. So they have all those methods of trying to understand what was there. This is another method, very cool one, because you can eat and drink the method. And I think we are helping them. And again, it was a dream of mine to be archaeology, to contribute something to the archaeology research beside my biology work. So I'm very happy that I made it, that I fulfilled it.